Good morning. My name's Chris Armstrong, Deputy Chief Scientist and Engineer. Apologies from Hugh Durrant-White, who's been whisked away to Adelaide to give a keynote speech today. But Hugh fortunately did have an opportunity to record a message before he left, so press play on that and we'll get ready to go. Thank you.
Welcome to the first New South Wales Science and Research Breakfast Seminar of 2021, and indeed the first in person for the last 13 months. I would first like to acknowledge the Gadigal people of the Eora Nation, the traditional owners on the land on which some of us at least are gathered today, and pay my respects to their elders, past, present and emerging. Welcome, as I said, to the first New South Wales Science and Research Breakfast Seminar of the year. Due to ongoing restrictions, audience numbers at Parliament House this year, we are bringing you the series from Fishburners in the Sydney Startup Hub. A calendar of this year's seminars is available both on your seats and on our website. For those in the room today, please maintain social distancing, ensuring you have scanned in and made use of the hand sanitizer provided. Those of you watching on live stream will be able to submit questions via the chat function on the right-hand side of the screen. Time permitting, these will be asked at the end of the seminar. The last breakfast seminar we were able to hold in person was on 26th of February 2020, when the 2019 New South Wales Scientist of the Year, Professor Rosa Mull of NSW Sydney, addressed us at Parliament House. We are all acutely aware of the COVID-19 pandemic that has prevented us holding a large in-person seminar for the last 13 months, and perhaps none more so than today's speaker, the 2020 New South Wales Scientist of the Year, Professor Eddie Holmes. Eddie is a global leader in research on the emergence, evolution and spread of viruses. He has a particular interest in investigating how viruses are able to jump species boundaries, occasionally causing disease epidemics and pandemics. In January 2020, Eddie became the first person in the world to publish the genomic sequence of SARS-CoV-2, enabling urgent work to commence globally on both virus detection tests and vaccine development. He followed this with fundamental research into the animal origin of SARS-CoV-2, helping to demonstrate the presence of related viruses in bats and pangolins, and showed that coronavirus can jump species boundaries and emerge in new hosts. It's no exaggeration to say that Eddie's work has been instrumental in helping the world get to the point where vaccines are now being rolled out and the future can be viewed with a degree of optimism. In Eddie's career, he has made major contributions to our understanding of the fundamental mechanisms by which viruses evolve and helped to pioneer the use of phylogenetic methods to track the spread of viruses within populations. His other major research themes are using genomics to understand the epidemiology of major human and animal pathogens, and revealing the extent and structure of global virus diversity, the so-called virosphere. His work has led to fundamental insights into the origin and spread of numerous viruses that have had a major impact on human and animal health, including hepatitis C, HIV, influenza, West Nile dengue, Zika, and Ebola. Befitting his achievements, Eddie has received numerous accolades. In 2003, he was awarded the Scientific Medal by the Zoological Society of London. In 2008, he became a Kavli Fellow of the National Academy of Sciences, USA. In 2010, he won the Faculty Scholars Medal in the Life and Health Sciences at Pennsylvania State University. In 2015, he was elected a Fellow of the Australian Academy of Science. In 2017, he was elected a Fellow of the Royal Society of London and won the New South Wales Premier's Prize for Science and Engineering in the Biological Sciences. As I mentioned, in 2020, he was also honoured as the New South Wales Scientist of the Year at the New South Wales Premier's Prizes for Science and Engineering. Now, I will be joining the seminar online, but I would like all of you in person, please join me in welcoming Professor Eddie Holmes from the University of Sydney as he presents the discovery and origins of SARS-CoV-2.
Good morning. As Hugh said, we've all been at home giving seminars over the last year. It's the first time I've given in person for over a year, so I may be a bit rusty. I've forgotten how to do this. So what I'm going to do today, thank you, Hugh, for that very generous introduction, is I want to talk a bit about, as my title says, how we found this virus and where it may have come from. And I've added the word and evolution, or words and evolution to my slide, because I'll also talk a little bit about where we are now, because we're hearing about how the virus is evolving and new variants are appearing. I just want to discuss what that kind of means for how we might handle this virus, okay? So that's the plan. Before I get to SARS-CoV-2, I just want to give you another story that I hope sets some of the key topics up for you. And it's the story of a different virus, okay? And I wind the clock back to 1978. Now look at me in the audience. I think most of you are probably alive in I might have to move this up. Okay, there you go. I suspect most of you are probably around in 1978. And this is a page from the Daily Mirror newspaper from the UK. And if you read it, it says, you can see that the time is quite dated because it discusses disco at the bottom there. Elvis is mentioned. He was a well-known entertainer, I believe. The top part, though, the top two stories are discussing quite a sad tale of a person who's just died. And she's there in a wedding dress. And at the top, it talks about a mystery of the smallpox leak. Okay. And what this story is referring to is the very last case of smallpox ever recorded anywhere on Earth. Okay. And bizarrely, that case was found in Birmingham, UK. And it's a story of a very sad story of a person called Janet Parker. She's the person who's in that wedding dress in that newspaper thing before. And she was a medical photographer at the University of Birmingham. And she was exposed somehow to a smallpox virus that they were working on in that building, in the University of Birmingham, that was being worked on the floor below her. So somehow the virus got up the floor, she got infected, and with smallpox, and she died. Okay, she died on it says there. I'm not sure which one to. If I can choose, I'm not sure I can actually point to it, but she died on September the 11th, 1978. Okay, and she actually ended up in this quite miserable place. You can see it there, the Catherine de Barnes Isolation Hospital, and that's where she spent her last days. It's a very kind of sad story. It's actually a very good book you can read by Mark Palin called The Last Days of Smallpox on this story. So I discussed smallpox very briefly because it is an extremely important virus in terms of human history for a number of reasons. As I'm sure you well know, it's the only human virus that to date we've eradicated from our species, and that was eradicated by vaccination. And of course, everyone knows the story of vaccination and smallpox. Every school kid knows this. It was basically first developed, not invented, but developed by Edward Jenner back in the late 18th century. And you all know the story of cowpox and the cow maze and that kind of thing. I won't go into any detail. And it's a kind of celebrity thing from the time. So I think that's a Hogarth painting you can see there of Jenner vaccinating a kind of rich person, 19th and 18th century. And the terms vaccine and vaccination actually come from the phrase variolae vaccinae, or smallpox of the cow. So that's kind of the history of vaccination. And I've worked with my colleagues for many years on trying to understand the origin and evolution of smallpox, as I say I'm going to discuss for SARS-CoV-2. And so, for example, what we've done is we've gone to various locations globally to try and find old smallpox samples. So, for example, here we go to a place called Vilnius in Lithuania. There's a map of Eastern Europe there, and Vilnius is kind of highlighted. And in Vilnius, there's a church, an old church is there. And in that church, there's a crypt. In that crypt, there are coffins. In those coffins, there are mummies, right? And some of those mummies are child mummies, and some of those mummies died of smallpox. I'm not going to show you the mummies because you just had your breakfast. It's a bit confronting, right? And what we can actually do is you can sample the lesions and smallpox from these mummies, extract the nucleic acid, what's there, and sequence it. And we did that, and we managed to sequence the complete genome of smallpox virus. The virus is actually called a variola virus, from a sample that's from 1650, so 400 years old. So that's at the time, that was the oldest known smallpox strain, right? So that's the kind of thing you can do with genomics. And once you've got that genome, you can then start to look at its evolutionary history. And that's what I'm going to show you for SARS-CoV-2. Now, the way we look at evolutionary history, just for those who are not familiar, is we draw things called evolutionary trees or phylogenetic trees. And just to think about it, it's exactly the same as a family tree. You all know about family trees and pedigrees. Okay, so here we have a family. We have some children and their parents, there's 2 generations. And you can just easily track the lines of ancestors and descendants in a family pedigree. You're all familiar with doing that. An evolutionary tree that I'm going to show you in a second, that you can see throughout this talk, they're exactly the same. They're stretched in time. So rather than 2 generations, they're thousands of years or hundreds of years. But it's exactly the same principle. Okay. So, for example, that's a kind of that's a family tree. An evolutionary tree looks like this. It's exactly the same, but I've spun it on its side, so you can't really see. So the base of the tree, if I can zoom this one here, that there, you can see the base of the tree. That'd be like the grandparents, and the tips towards the present, they'd be like the children. So it's exactly the same thing. And this tree is not a random tree. That's a tree of the pox viruses that include smallpox. And if you kind of look, it's very hard to kind of point to it from here, but you should see a yellow circle there. That's the smallpox virus. That little cluster, that little cluster of brothers and sisters is smallpox. And you can see it says Lithuania. So my Lithuanian strain falls in that little cluster. And you can see all around it, some of the names you can just, as it's written on the slide there, it says cowpox, vaccinia. And the idea is these are all animal viruses. Okay, so Jenner very famously used cowpox. Now, one of the questions we were interested in is, okay, so during Jenner obviously started vaccination. During the 19th century, there was quite a big vaccination campaign against smallpox, right? Which we're now entering for SARS-CoV-2. What vaccine strain did they actually use? When they vaccinated people with smallpox back in the 19th century, late 18th century, what were they using? So what we actually managed to do was find from a museum in the US some old vaccination kits. So these are from the US Civil War. See the picture there. They date to about 1860. And in those days, there wasn't a needle that you get at your doctor. It's like a little pad that had the vaccine and a little knife. You can see the knife there, a little strip. And so basically, they cut you and they rub in the vaccination. But it's a lot better these days, right? They rub the vaccine in, and that would be your vaccination. And what we managed to do, we took those kits, and if you wash them in water, the sediment comes off, the residual comes off, and that actually contains the vaccine strain that they used in 1860, right? Hidden in there. And we sequenced that, and that turned out to be, where the blue circle is, you can see it on there, a thing called horsepox. Okay, so the vaccine that they used was not cowpox or vaccinia, it was a thing called horsepox, right? Now, horsepox, all these names are actually very misleading. Horsepox is not from horses. It was found once in a horse from Mongolia, but we don't know where it comes from. Cowpox is not from cows, right? It was once in a cow in Gloucestershire in 1798, but it does not necessarily come from cows. And so what you're seeing here, we have all these pox viruses. They're animal viruses. We don't know exactly where they come from. It's the same story as you can see with SARS-CoV-2. Are they bats? Are they rodents? The animal kingdom carries lots of viruses, and occasionally they jump into humans that cause disease, like smallpox did back then. Okay, it's the same thing. Okay, so I raised smallpox. Oh, sorry, so we're now actually going on to try and find more about the origins of smallpox. Now, I mentioned already, as you all know, the story of Jenner very quickly. So he had this very famous cow that he used to get the first cowpox extract. That cow is called Blossom. Okay, it's a kind of celebrity cow. So it was painted by, I think, again, this is Hogarth, it's the Instagram of the day. So this is Blossom, very famous cow. And amazingly, the hide of Blossom is still around, okay? So if you go to St. George's Hospital in London, If you go to their library, there's an exhibit about smallpox vaccination, and behind that glass cabinet is Blossom's hide, or supposedly Blossom's hide, right? If you look, you can see the horns look kind of the same. It's a kind of white bit on the head there, you see. Kind of looks a little bit like the painting. It may or may not be Blossom. We've actually managed to take a swab of Blossom, this hide, to see if we can sequence it, to see does it actually have a virus in it that might be like cowpox. Now, okay, this hide, as I say, it's in St. George's Hospital in London. It's behind a glass screen. And that glass screen is there to protect it from kind of noxious things in the environment that might kind of float around, okay? I had to rehearse that joke. I've laughed. Okay, so... anyway, and so that's Blossom. We've also been trying to get hold of even an earlier celebrity smallpox victim, and that's the Pharaoh Ramses V. And the story is that Ramses V, very famous, this kind of textbook stuff, Ramses V may have died of smallpox. And that was from the very old 20th dynasty in Egypt. And we've actually got, again, samples, we think, from Ramses to try and see, did he really die of smallpox? Okay, so I mentioned smallpox for a couple of reasons. First, it's a classic case of emerging disease, okay? And it's one that's come from animals to humans. And actually all examples of disease emergence, like smallpox or flu or COVID-19, they're all about pathogens jumping from animals to humans, okay? It's the same thing. I also mentioned because, as you would have been aware, the Janet Parker story is also a story about a laboratory and laboratory escape. And I'll get to that with COVID-19 shortly. So smallpox and SARS-CoV-2 are actually examples of there are many of these emerging diseases. You can find online these maps of the world that show you where these jumps of a pattern from animals to humans have occurred in recent years. And there are a myriad of these. In Australia, of course, we have Hendra. We've had Ebola in Africa, Zika in the Americas, flu comes from Asia too. And there are lots and lots of them. And it's kind of hit the popular press. Of course, we had the movie Contagion. I guess that'd be Contagion 2 now coming out, who knows? There's Contagion. There's a book called Spillover. If you actually want a good general book about disease emergence, Spillover is very good. It's by David Crum. I'm actually in it, but apart from that, it's actually pretty good. That's a good book. And what I do for a living, normally, apart from jokes about Boris Johnson, is what my group do is I try and understand how diseases emerge. So I take the natural world and I look at the places where humans and animals interact, because that's the fault line. If you take a very simple analogy from earthquakes. The tension point where jumps occur are where people interact with wildlife. Okay, that's where viruses can jump. So what we try and do is we sample across that natural space. So, for example, people who work with livestock, or where human society have encroached into green belts and they're exposed to animals, what's jumping between humans and animals? So what's in the animals and what's in the humans? Okay, what moves across? So those kind of red lines on that kind of figure there, that's where we sample. And then we take a sample and then we use genetics to work out what viruses are there. I won't go into the genetics in any great detail. Suffice to say, we use a technique, it's a very simple technique. It's got a fancy name, it's very simple. And it's called meta-transcriptomics. So you can imagine, let's, for example, take one thing I'm working on is, which you're familiar with, is tick-borne disease in New South Wales. So people get bitten by ticks sometimes get disease, you may have had it yourself. There's been a big question, what's the causative pathogen? What's doing this? And so what you can do is you can take what we do is we take blood samples, skin biopsies at the kind of lesion, okay, we were bitten by a tick, and then we sequence all the RNA, not the DNA, the RNA. RNA is the kind of information molecule in the sample. And the RNA is all the genes that are expressed. So all the genes expressed in that tick bite we can sequence. And the genes that are expressed come from the host, so your genes, right? Your immune genes, for example, or from the bacteria, or from the virus, or fungus, or parasite. And all those little pictures there are showing the different pathogens that could be in those samples. So we sequence all that, and then we try and work out what can we see. Can we see a pathogen in there? Can we see a new virus or bacteria? Okay, that's the technique. And that's the technique that we use to discover SARS-CoV-2. I'll discuss that in a second, right? This is called meta-transcriptomics. It's basically sequencing all the RNA and using computers to work out what it means. And one thing you get from doing that is the number of viruses out there is just absolutely astronomical. And what this kind of very simple figure here is just trying to show you is how big that unknown universe of viruses, or the virus sphere, actually is. So the small dot at the top are the number of human viruses that are known. It's about 200, 250 or so. The slightly bigger dot is how many viruses are known throughout that are classified. It's now about 6,000 or 7,000. And the big circle is the bit that's unknown. And you can see that we basically have scratched a tiny, tiny fraction of the viruses. Viruses are absolutely everywhere. If you go and sample wildlife in Sydney, you go to a food market, the plants that you buy, the food that you buy will have viruses. They're everywhere, okay? So it's a fact of life. Okay, so now that's the kind of background. Now I want to get on to SARS-CoV-2. So for a number of years, so when I do this the core of my work is going out there, like I say, and looking at places where humans and animals interact to see what viruses are there and what could jump host species. And a lot of work I do, excuse me, I'll get a drink. A lot of work I do is in China, which is actually a very good place to work on this. So for example, here I went to, this is some photographs from the Zhejiang province in the southeastern China. And I went there a few years ago. And it's kind of these very old villages. You can go to kind of steppe landscape. And then they take you there. Then you walk around a kind of mountain for a couple of hours. And you get to a cave. And then the people have to put their PPE on. You're quite familiar with that now, okay? You put your PPE on. And then and unfortunately, you have to go into these caves. That you're seeing there. I had to go into that myself. There's a cavern you're seeing there. And then in these caverns, there are bats. Lots and lots of bats. And so our group and many others, for a number of years, have been sampling bats in China and in other locations. In fact, I've done it in Australia as well. Okay, and then if you use that metatranscriptomics, you sequence all the. We would sometimes take fecal samples, sometimes tissue samples. Bats carry one hell of a lot of viruses. They're absolutely full of viruses. Partly because their immune systems may be different, partly because there's a lot of bats. They can carry lots of different things. So here's a paper we wrote just in 2007, I think it was 17 maybe, and it's just showing, it's called the diversity of bat viruses in China. And there's lots. And so we were already sampling these bat viruses from China. And we wrote in that paper, and this is not trying to sound profound, but we wrote that, these things could emerge, right? There's a lot of viruses in bats, a lot of viruses that look kind of potentially dangerous, like coronaviruses. So there's a chance that, this could happen. We could get an epidemic. And so COVID-19 is of absolutely no surprise to me whatsoever. It was clearly going to happen because we could see the viruses out there. We knew they were jumping. It was an accident waiting to happen. Right, so that's then wind forward a little bit. It gets even closer to the real thing now. So then, as well as Zhejiang province, we've been doing a lot of work in other parts of China. And over the years, one of the locations we'd also sampled a lot in was Hubei province, in the kind of middle of China, where the Yellow River runs, right? And we'd sampled lots of wildlife around the city of Wuhan is in Hubei. So we'd sampled lots of bats and insects and mice around Hubei. But we'd also sampled patients in the city of Wuhan. And in particular, we went to a hospital here called Wuhan Central Hospital. And so when you saw Wuhan last January, this was the hospital you were seeing. This was the epicenter of the epidemic. And I was there in 2016. You can read it. It says my name's there in lights. It basically says, welcome, you're giving a talk, I think. I hope that's what it says anyway. That's Wuhan Central Hospital. That became very famous. And what we were doing is this sounds quite prophetic, is we were taking people who had pneumonia and acute respiratory disease to see what microbes could be infecting them. This is prior to COVID. It's actually 2 years before COVID. We wanted to know. There are lots of pneumonia people get very often, okay, and it's often undiagnosed. So people, so it's a very common disease. The question we were trying to ask was, what's responsible for pneumonia and his acute respiratory disease in Wuhan? We were doing this 2 years prior to the outbreak, okay? And it says that we had about 408 patients, plus loads of controls. And we were taking a sample called, the technical name is a bronchoalveolar lavage, or it's a lung wash. And so that's quite a good sample looking for respiratory disease, okay? So taking these lung wash samples and then doing that metatranscriptomics to see what these patients had in this hospital in Wuhan a couple of years ago. And so this is a bit technical, I apologize, but basically they had a ton of stuff, right? As you might expect, there's bacteria, there's fungi, there's viruses. There was nothing novel. This is 2016, 2017. This slide of the grey kind of slides, that's a heat map. It's just showing you some of the pathogens. It's coded, but the pathogens they had. Basically, they had lots of common cold coronaviruses, influenza, parainfluenza, respiratory viruses, a whole bunch of the standard stuff you'd expect. They'd had a whole bunch of bacteria you'd expect. There was nothing novel at all. Critically, there was no SARS-CoV-2, right, or the first SARS virus. That wasn't there at all. Okay, so that's 2 years prior to the outbreak. We stopped sampling in December 2017. There's nothing there. And the evolutionary trees on there, you're just seeing, are showing you the viruses that we discovered and where they fit. And what you can see just from those trees very clearly is that they're linked to kind of global sequences. So Wuhan is a really global place, a big city, middle of China, very well connected. And so it's getting kind of global mix of viruses, right? Because it's a hub, a real big hub. In fact, the only thing that was unusually found, you can see it there, is a zoonotic pathogen. It's a bacteria that causes psittacosis. Psittacosis is often called parrot fever. And people who work with birds sometimes get psittacosis. We have one person who had pneumonia because they got this parrot fever, okay, which is kind of strange. That was the only thing that was unusual. There was nothing else there. But the key thing was we were on site in Wuhan collecting these respiratory samples in the hospital in Wuhan prior to the outbreak, right? So by chance, we were kind of on site, okay? Then, of course, roll forward to the famous bit, right? It's the end of 2019. The first I heard about this outbreak was on a website called ProMed. And ProMed is a really great free tool you can get. You can download it on your phone. It's on Twitter. It's on the web. And it just gives you a daily update of outbreaks of disease globally. It could be animal disease or human disease, anything. And so this is the actual original ProMed post. I saw this on on New Year's Eve, New Year's Eve 2019. So December 31st, 2019. This is what was up there. And you can see, if you can read it, it basically says there are 4 cases of pneumonia in China associated in Wuhan, associated with a seafood market. Now, I've been to that market, and I'll discuss that in a little while, okay? But I was immediately interested because it was pneumonia in Wuhan that I was working in, and a market that I'd been to, which I'll show in a second. So I thought, okay, this is really cool. So at that point, we were not worried, okay? So then I contacted my friend and colleague, Professor Zhang in Shanghai, to see, was he going to work on this? And what happened was, a few days later, on January the 3rd, Professor Zhang, who's in Fudan University in Shanghai, was sent 7 lung wash samples, because again, that's the sample we were working on, to his lab in Shanghai to sequence. And these 7 lung wash samples were all people presenting with pneumonia, and they were all people associated with this seafood market, and I'll come back to that in a second. So then, that's January the 3rd. On January the 5th, so 40 hours later, he managed to sequence all those samples, okay? Of those 7, one of them had really lots and lots, using a metatranscriptomics, that approach we use, right? Lots and lots of hits, of matches, of sequence to SARS-CoV-2, okay? So it took 40 hours from the virus, sample arriving in his lab to us to sequencing the virus, 40 hours. It took, put in context, it took 2 years to find that HIV caused AIDS. 2 years, right? So this is quite extraordinary what we can now do with modern genomics, so 40 hours. And that sequence was finished on January the 5th, 2020. And Zhang and I were talking on the phone that day. And then on the same day, it's very important, he wrote to the Ministry of Health in China and said, basically, this is what it is. It's a new coronavirus. It looks like the first SARS coronavirus. It's very similar to the one of 2000, 2003, remember? It looks like that. And because it looks like that, we knew automatically it was going to be respiratory, right? Because coronaviruses are respiratory, okay? That absolutely is a defining feature. And so Zhang, we discussed it, and he wrote a letter to the Ministry of Health, and it says, this is likely a respiratory pathogen. It's likely to be dangerous. People should take precautions. Okay, that was the same day. Now, I don't want to get political about this, but one of the things that really didn't, I think one of the unfortunate things that was done is that human-human transmission was not officially recognized until about January the 20th. So there's a 2-week gap between the virus being sequenced by us and the authorities realizing that there was human-human transmission. But to Zhang and I, it was obvious from day one. In fact, we have emails and we're discussing, is the transmission asymptomatic or symptomatic? Okay, because it was obvious, it's a coronavirus. Coronavirus are respiratory. So unfortunately, the ball was dropped for a bit there. Okay, so that was it. Now, I should also say, we were the first people to release the sequence. We were not actually the first people to sequence it. Prior to us, there were a number of other groups that were, this is all now quite famous stuff you can find online, other groups that have been sequencing the virus. And the first actual sequence came from a company called Vision Medicals on the 25th of December. So these doctors in Wuhan saw they had something. Their diagnostic tests weren't working. They didn't know what it was. So they sent it quite reasonably to a local sequencing company and said, can you figure out what this is? And that company first got some data. And this is from the, on the right there, you're seeing what the original results. They first got the data, and they sequenced it on 25th of December, and they got hits for SARS virus, okay? And they basically went, oh, beep, it's SARS back again, okay? And that went all over social media in China, that SARS was back. And of course, that was a toxic thing. People didn't want to have SARS back. And it was all, and that caused pandemonium. Then a second company confirmed that and resequenced it. Now, when they said SARS was back, what they couldn't see was it wasn't exactly SARS. It was related to SARS virus, but actually it's a different virus. So there was some confusion early on. But there's a lot of toing and throwing in the late December, the last week of December is when people really started getting a number of groups of sequencing, and we were one of them. Okay, though. But as soon as you had the first sequence, it was, and I wondered in detail, we knew exactly what it was. It was pretty obvious this was a coronavirus and related to SARS-1. This is one of the first trees that we did. You don't need to kind of see it very closely. But at the top in red, that's the first SARS coronavirus, right? And a bit further down in red, that's the new one. And it says 2019, halfway down, it says 2019, little human symbol, NCOV. That was what we called it then. And you can see that the two red bits are quite similar. So we knew right from day one it was closely related to SARS virus. We knew that. And that's why we said it's respiratory. Also, at the bottom, you can see some other human symbols. There's some other coronaviruses that infect humans. So we knew it was a kind of human coronavirus. Critically, the ones in the branches in black, they're all from bats. So we knew right from the start it was like the first SARS virus, but a bit different. Also, it was related in evolutionary terms, okay? Its family tree had bats very close. So we can see that right early on. The genome structure, I don't want to talk about in any great detail. That map you're seeing there is just the genome sequence, kind of schematic, what it looks like. There's one little bit of it. So if you remember, you all heard this now, the virus kind of looks like a big ball. On the ball, the outside, there's a protein called the spike protein. And that's the bit that attaches to the host cell. And there was lots and lots of interest about the spike protein. In particular, the spike protein had some little bits of sequence that looked quite unusual. That bit of sequence is called a polybasic or a furin cleavage site. So it's actually 4 amino acids, 4 bits of sequence that are unusual in SARS-CoV-2. I'll come back to that a bit later on. So again, it's a standard coronavirus, and we could see it was like the first coronavirus and a bit like bat viruses. Right, I'll come back to that. So then the big question is, where does it come from, and what's the animal reservoir? And I've already told you, right from day one, we could see it was closely related to viruses in bats. That was very, very obvious. And it's not just any old bat, it's particularly horseshoe bats. So you can see the picture of the horseshoe bat there. See the nasal structure? It looks like a horseshoe, right? So it's kind of obvious why they're called horseshoe bats. And they're actually from a genus called Rhinolophus. And it turns out that horseshoe bats are very common reservoirs for coronaviruses in China. They are full of coronavirus, okay, very, very commonly. So that was not a surprise. What was a surprise, it's kind of blown my mind, is that pangolins also have a related virus. Now, no one was expecting pangolins to have a virus. No one had worked on pangolin viruses ever before this sort of work. Right? Pangolins are nocturnal, kind of solitary animals, massively endangered because they're trafficked all the time for their scales, and their scales are used in traditional medicine. And what happened was the customs authority in China, in 2 provinces, Guangdong and Guangxi, had confiscated these pangolins that were being smuggled in because of their scales. And these pangolins were sick. Okay, they were not healthy pangolins. So the customs authorities alerted the vets to say, there's something wrong with these pangolins, what's going on? And so my colleagues then did genomic sequencing on the pangolins, and bizarrely, they have a virus that's very closely related to SARS-CoV-2. You would not have predicted that, okay? You can do evolutionary trees, and I apologize, a little bit complicated. You don't need to show us any detail. These are just different genes trees you're seeing here. If you look at one there, hope you can just see the little human symbol. That's the human virus. And the ones close to it in different colors, they're all from different bats. And there's bats in China, there's bats in Cambodia, there's bats in Thailand. All have viruses very close to the human strain. And a bit below, see this kind of pangolin symbol? They're a bit further away, okay? Quite extraordinary. The pangolins, although they're a bit further away, there's one bit of sequence called the receptor binding domain, and that's the bit of the virus that absolutely attaches to the host cell. So the virus binds the cell to get in. Where it attaches, that bit of sequence, bizarrely, in the Guangdong pangolins, ones confiscated by Guangdong Customs, it's almost identical to the human sequence, kind of very strange. So, bats have it, pangolins have it. But there's a gap, there's an evolutionary gap. So I can't really show you very clearly, but if you look at the tree, you've got the human virus and the closest animal ones, and there's a bit of a branch between them, some distance. And it looks very small on these trees, but in evolutionary terms, it's about 20 years or so of missing evolution. So you have a human virus, the animal viruses, and there's something in the middle we haven't sampled yet. And we don't know what that is, okay? I'll come back to that in a sec. Now, the bats, a lot of interest in bats, and they do carry an awful lot of viruses, and it is quite startling to see how many viruses they cover. So we just did another study, and my colleagues went to a tropical botanic garden in Yunnan. I can't say that word, I apologize, the first word, but it's a beautiful tropical botanic garden in Yunnan province in southwestern China. And in this garden, and it's only about 1,100 hectares in size, in the bats in that, and they're not in caves, they're in kind of like, trees. These bats, we managed to sample 26 new coronaviruses, novel, completely new, in a 1,000 hectare space. So you can just imagine the number of viruses that are out there in nature, the number of coronaviruses, is absolutely astronomical. That tree is just showing you some of the diversity. And in fact, if you actually look at these rhinolophus bats, this is just showing their distribution across Asia. The kind of yellowy colours are where you're seeing the bats. They are all over Southeast Asia. So this part of the world has lots of rhinolophus horseshoe bats, and they're going to have absolutely, buckets is a technical term, buckets of virus, absolutely, okay? Because there's amazing diversity of viruses out there, just huge. So bear that in mind. Pangolins, again, I mentioned them very briefly. So the one thing that people haven't really cottoned on to pangolins is they were ill. They were not healthy animals at all. So what you're seeing there, I apologize for one slide there, I know it's not very pleasant. That's some necropsies of a pangolin. They were diseased. The top there, these are CT scans of a pangolin lung. I bet you never thought you'd see that before. So the white one on the left is the sick pangolin, the kind of the one with more grey on the right is the healthy one. So they're ill. The pangolins were ill. Also, this is a very technical thing, but that little picture there, that's showing you the gene expression pattern. So basically, when a human has COVID-19, various immune genes are turned on when you're sick. The same genes are turned on in pangolins, it turns out. So it's a very similar disease presentation. So pangolins get a very similar kind of COVID thing as humans do. Okay, so as I said, there's a gap, okay, between the human ones and the closest animal ones. And the question is, what goes in that gap? And what the WHO have just announced in their report, thank you for timing that for my talk, by the way, very good. What they've just announced is they think the most likely scenario for how this virus emerged is it comes from bats through an intermediate host to humans, okay? So the question is, what's that intermediate host? What is it? And the animals that I think are most likely are mink and raccoon dogs. So mink, I'll show you a raccoon dog in a second. Mink, you all know. These animals are fur farmed in large quantities in China, okay? They're wildlife that are farmed. And I think they're pretty likely to be intermediate hosts. Mink, we know, carry the virus. The virus in Europe has gone from humans to mink, and then from mink back to humans. So mink are a really good source of virus. The other animal that I think is very likely are raccoon dogs. They're my personal favorite. They look kind of shifty, right? And I say that because in 2014, I went to this market, this seafood market, right? And I'm going to show you some photographs. It's a pretty confronting place. It's kind of sweaty, it's steamy. There's lots of internal roads, okay? And there are lots and lots of animal stores. And this is a map that WHO have just produced of the market and the different stores. And each little block is like a different market trader. And the ones in colour are the ones they've tested for virus. And you can see the ones in the kind of orangey colour, they are the places where they found positive swabs. So what they've taken a swab on a surface, be it a wall or a bench or the floor or a grate, and they found virus on those surfaces. And you can see they're all clustered on one side of the market, on the west side of the market. And that's the side that the animal traders really were. The fruit and veg people are on the east, the animal people are on the west. It's the west side that has the virus. So there's clearly mass transmission going on in that market, absolutely. Now, we don't know whether it's going from just between humans or whether animals were involved, but there was lots and lots of transmission. And when I was there, the key thing is I took photographs of wildlife in that market. I was there in 2014. And I took photographs of raccoon dogs. Okay, so that's a raccoon dog. They're kind of very strange. They look like kind of photoshopped animals, but they really do exist, right? And so these raccoon dogs, they're my photographs, the two on the left there. They were in cages stacked up in the west side of the market. And that's where most of the positive environmental samples were. So I suspect that's a really important avenue for this virus. My bet is on the farmed wildlife trade being brought into this market. That, to me, is the most likely scenario. And that's what WHO have decided. We're lacking the smoking gun or smoking raccoon dog. We don't quite have it yet, but I think, and tellingly, these animals have not been tested in China, nor have mink. Okay, and I suspect they're the most likely. Okay, I'm just going to skip that one because I've got time. Right, so then now the million-dollar question. So what you've also heard lots about is, did this virus escape from a lab? And this is kind of growing in momentum. I just want to address this very quickly. And of course, the reason why that's a topic of conversation, and something that I thought about immediately as well, so it's not a kind of strange thing, is that in the middle of Wuhan also is a very big virus lab called the Wuhan Institute of Virology. And they were working on bat viruses in that lab. And one of the closest relatives to SARS-CoV-2, called RATG13, is from that lab. And that's led to an enormous amount of debate that's kind of been grown, and very lots of politics. But the critical thing is actually look at a few facts, right? So here are the facts. So I'll just go through them. One, and sorry, this is a list, right? So one, there is no evidence that SARS-CoV-2 is engineered at all. And here I heard people talk about covert military operations. Absolute nonsense. I'll say it on camera. Nonsense, right? There's no evidence of engineering whatsoever. The idea that Chinese military are working in a civilian lab and let them publish academic papers on the virus they're then using as a bioweapon is just mad. So don't believe a word of that. This bat virus, RATG13, is not the direct ancestor of SARS-CoV-2. It's a relative, but it's actually not that close. There are other viruses that are closer in other bits of sequence. And in fact, if you look at nature, all the component bits of the virus are found in other natural species. This little thing here is the furin cleavage, like a little bit of sequence. The top one's SARS-CoV-2. The one below it is RATG13. You can see I can move this. There's a gap there, but in other viruses, the gap is filled, right? So there's nothing unusual in that sequence at all. Therefore, for it to come out of a lab, they'd have to have be working on a secret SARS-CoV-2 virus that they've not told anyone about that's closer to SARS-CoV-2 than RATG13, right? So why they would do that, I have no idea. They published all their viruses in the past. There was no reason not to publish it, before the pandemic. But they claim it's not there. And frankly, I believe them. So that's where we are. SARS-CoV-2 is not necessarily from bats at all. As I said, they're probably an intermediate host. And it's not necessarily from Yunnan. Yunnan is just a place where they do lots of sampling. That's not necessarily where it comes from. Just that Yunnan's a great place to go and sample bats. There could be sampled bats anywhere in China, but we happen to sample a lot from Yunnan. I've heard talk about SARS being perfectly adapted to humans, and that's kind of unusual. That's really not true either. So it does do very well in humans, but also it actually does well in many other organisms. This is a technical point here from a paper in Nature. And what they're saying is basically the pangolin virus on itself works really well in humans. Okay? So you didn't need to adapt this virus to humans. It was going to work already. The ones from nature are very, very good human viruses, particularly the pang. That's what they're saying there. If you look at the maps, the first cases in Wuhan are close to the market, and it spreads away from there. It doesn't get to the Wuhan around the astrology for a little bit later on. So the spatial distribution fits the market first, then the lab, quite clearly. And finally, there's no evidence. So the people with astrology claim that all their staff are PCR and antibody negative to the virus. Okay, so no one's infected. So if that's true, that rules it out automatically. So either this is the biggest cover-up in history and they're all lying, right? Or there is no evidence at all. I'm very prepared to believe it's out of a lab, but give me the evidence. At the moment, there is absolutely none, okay? But this is going to go on for a very long time, I can absolutely assure you. Right, so 5 minutes. 5 minutes to talk about where we're going now. Just very quickly. So the virus has then emerged. We're now sequencing it frantically. You've all heard about that. We have now sequenced something like 900,000 genomes of this virus globally. Just extraordinary. Pretty soon, we'll have 1 million genome sequence, okay, within the next couple of weeks. Just incredible. And the virus is gradually evolving. It's actually picking about 2 mutations a month on average as it evolves through populations. I haven't got time. I'm just going to shoot through this. So what we've been doing is trying to classify the virus as it spreads globally. And we developed a very simple online tool where you can take your sequence, your virus, and you can work out what lineage it is. So you take the tree of the virus, you can divide it into lineages, okay? And that's what I'm showing there. We're called very technical names, A, B, C, things like that, and A1, B1. It's a very simple lineage classification system. And this little tool called Pangolin, pretty clever, hey? With a really great logo, I'll say so myself, that allows you to find out what lineage your virus does. Now, we've classified something like 887 lineages so far, but there are 3 of them you're hearing a lot about in the news at the moment, and they are the so-called lineage or variants of concern. And there are 3 that you want to know, and the technical names are as follows. One is called B117, and it's often called the UK variant. It's not. That's because it's found first in the UK, I should say. We call it BB117. That's the one that's now the outbreak in Queensland, is B117. One's called B1351. That's the one that's found in South Africa. And there's one called P1, that's from Brazil. They're the 3 that people are most worried about because they're spreading very quickly. And I'll discuss B117 in a second. This little picture here on the right, that's the genome map, and it's showing where the mutations occur in those 3 variants. And they're mainly clustered in the spike protein, the kind of orange thing, right? Now, the key thing is all those variants are characterized by lots and lots of mutations. Just loads and loads of mutations, very unusual. And you've heard about these mutations. And the key thing is the same mutations are appearing in all these different variants. And to make it easy, we've given them kind of stupid nicknames in the group, right, to make it easier, because they're so complicated. So, for example, the first mutation we saw was an amino acid 614 in the spike protein. It was a D amino acid to a G, so asparagine to a glycine change, right? D614G, so we call it Doug, right? DG, Doug, right? We've also got N501Y, we called it Nelly. And you can see the name, sorry, I apologize if you're one of these names, right? The mutation that looks worst is one called E484K. Originally we called it Eric, EK, right? We've now thought this is actually the worst one, so we called it Eek because it's kind of bad, right? And the key thing is the same mutations are appearing independently in these different lineages. And so the big question is, what's driving this? I really haven't got time to, it's a bit late, I've got time to explain this, but basically, there are a variety of theories for why suddenly all these variants of concern are appearing. What's going on? Is it, and here are the different theories there, is it because the lockdowns we've had are selective for different variants, okay? Is it immune escape? Is it increased transmissibility? Is it people have chronic infections so they don't clear the virus? Are they generating the virus? I suspect it's the top two, it's immune evasion. So as the virus has spread globally, people are gradually getting immune and now vaccinated. And that's putting selection pressure on the virus. And I suspect all these variants are actually in some way immune escape. It could be interferon escape, we're not entirely sure. I suspect that's what's going on. And this is going to happen. The more immunity rises, the more selection there's going to be on the virus. It's going to happen, right? And we will likely have to update the vaccines. Now, of those three, the one that's most concerning is B117. That's the one that's from the UK. And that's the one that's now broken out in Queensland. And it's concerning for two things. First, it does appear to have an increased transmissibility compared to other variants. I'm sorry this is just taken straight from the paper, but you can read it, what it says there. It says, I've highlighted, there's a 43 to 90% increase in transmissibility of this variant, right? Compared to the original. So it's much more infectious. Okay, so that is a worry. Even more of a concern is the hazard of death, which is a nice way of saying mortality rate. It's between 42 to 80% higher if you have this variant. So it spreads faster and it's more serious. And you actually look at that table there, it's giving you mortality figures for different age classes. And people over like 80, the mortality rates are really high, over 15%. So this is a concern. Now the good news is, as far as I'm aware, the vaccines that we've got, mainly, although they don't protect against transmission always, they are very good at stopping death and serious disease against all the variants, including the UK one. So the vaccine is critical. It should massively reduce the mortality. It may not stop transmission, but it will definitely reduce mortality. So take the vaccine. And it's not just the UK now. You're seeing, this little graph is showing how this virus is spreading. And there's absolutely no surprise. It's spreading all over the place. It will become the dominant strain globally, absolutely. And I'd say, luckily, we can vaccinate against this strain pretty well. And so people in hotel quarantine that come into Australia, Sydney, whatever, Brisbane, Melbourne, well, not Melbourne, they are now very commonly B117. So it's no surprise that's the one that's escaped, because that's the one that's spreading all the place globally. But like I said, the vaccine should control it. Right. I'm just going to skip on to the last bit now. So how can we stop this happening again? Because I'm running out of time. I think there are three things we need to do. I think emergence, if I try to show, is all about human-wildlife interaction. Okay, that's what it's about. Somehow we need to stop that happening again by monitoring, reducing our exposure to the wildlife trade, live animal markets, better zoning. That's the fault line. That's where we get viruses from. Okay, so not building on areas where bats live, for example, okay, like councils tend to do. We need to stop doing that. We need to have much better surveillance mechanisms. There's still pictures in the middle of it. I'm showing you how you can do that. And there are good ways we can now use genomics and serology to survey for these viruses. So I think people who work at the human-animal interface, so in abattoirs, in live animal markets, in the animal trade, they need to be screened absolutely regularly, okay? Because they're the front line, they're being exposed. If we can survey them and detect them early, then we can stop the next thing happening. But that also requires data sharing and politics to get out of the way, and unfortunately politics is undermining this at the moment. And finally, the kind of critical thing, the picture on the left, we need to develop universal, which is very easy to say, very hard to do, universal vaccines or cross-protective vaccines and antivirus that recognize all the viruses out there. There are certain ones that always jump. Coronaviruses jump, influenza viruses jump. There are a few other families that jump all the time. We need to make now vaccines and antivirals that can protect against a broad range of those. We've been trying for a few years to make a universal flu vaccine. The same thing needs to happen for lots of other viruses too. That's the kind of Manhattan project, or the Apollo project, I should say, for virology. That's what we need to do. Massive investment. That's where science needs to be. Okay. I'm going to stop there. Lots of people are involved. I haven't got time. I'm running it over, so I need to just... I thank them all. I'll leave their names up. Thank you.
I've got time for a few questions for people that can stay. Any questions? Over here.
So with these new viruses, can sewage testing be used to detect new crossover, new pathogens which cross over from humans to animals, since not everyone's going to get tested or going to be eligible for tests?
Yeah, no, it's a good idea. So the question is, can you use sewage testing, wastewater testing? You can, if you know what you're looking for. The problem with sewage testing is the virus is very, very fragmented when you get there. So doing a simple PCR test, that's what you use now to test whether you're coronavirus infected or not. That works okay. To do the genomics that I do to find unknown viruses, it won't be as good because the material is so complicated and broken up. So I think it's definitely a useful thing, but I think it can track. If you know something's there, it can track it. Finding a completely novel thing at the moment, I think, is a bit harder.
I guess the simple question to me is, where does it end? Fauci tells us that the only way out of all this is the vaccine. We hear we have to build up a herd immunity, but we isolate, so we're not building up herd immunity, but we need the vaccine. But you look at the Spanish flu, you look at the Hong Kong flu, they eventually burn themselves out. But everything we hear indicates that this is going to go on forever. What's your opinion on this, please?
Yeah, I think this will become an endemic human virus. There's no doubt whatsoever. And flu has never died out. I mean, flu is an endemic thing. We've had it for years. My guess is the virus will gradually evolve into a seasonal respiratory infection, and that we'll need to vaccinate every year, right, or every 2 years, as we do for flu. There's no way, eradication is impossible because there's no way every country in the world will roll the vaccine out. It's just not going to happen, right? And the virus will evolve around it. So I think it'll be an everyday part of our lives, and we'll just have to get used to being vaccinated. Luckily, like I said, although these variants, or scariants as they're often called now, are coming up all the time, and the vaccines have different efficacy against them, okay? The good news is they all seem to control mortality pretty well, right? So you get the vaccinated, you might get a bit ill, but it should stop you getting a serious lung infection. So it's going to be part of our lives, right?
Question up the back in the blue first.
Look, I was just wondering, could you give us an indication of what your feelings are on the apparent claim of the Chinese government that there have only been about 4,300 deaths due to COVID-19 in China?
Am I on camera? There's a lot of politics in this thing. I think there's a number of countries where the numbers claimed may not be real. When I think in Wuhan, the modelling suggested very early on the number of cases in Wuhan was far higher than reported. Okay, I mean potentially hundreds of thousands of people in Wuhan were infected. So I don't know the data, but I think a lot of these things you need to be very careful about. I'm going to dodge your question in a very careful way. There you go.
Thank you. I'm enjoying the splendid isolation of Australia, and it seems that we have a zero tolerance to COVID coming into the country. This is great short term, but long term I can't see that it's a feasible position to take. What do you think it will take for that position to change politically?
Yeah, look, at some point the federal government will decide enough people have been vaccinated that they will open up the borders. We can't be in isolated like this. We've done extraordinarily well. I think there's a case that New South Wales has done better than any place in the world, quite frankly, because we've had 56 deaths from COVID in New South Wales. Extraordinary, okay? And we've not had a really hard lockdown. So I think we've done better than anywhere, really extraordinary. But it won't continue. My guess is once they've got to the level of when most of the adults over a certain age have been vaccinated, I think that's when they'll start to open up. The kids, I don't think will come into it. I think they might leave those alone a little bit. I think they're looking for a proportion of the adult population to be vaccinated. What the proportion is going to be, I don't know. Unfortunately, the vaccine rollout's not going as quickly as I hope it would. I think they've been too slow. Okay, I was hoping by the end of October, but they're now kind of hedging their bets. So it's hard to tell. It really is hard to tell. But I think they'll have to come out certainly by the start of next year, if end of this year. I think it has to happen. I can't.
Got a couple of questions from online. I might just borrow the mic.
Yeah.
We've got a question. What is the evolutionary role of the furin cleavage site? How did it get inserted into the SARS-CoV-2 virus?
So this furin cleavage site, this has been the most contentious, 4 amino acids of such absolute contention. And so there's been an argument that's been deliberately inserted. If you look at viruses, lots of them have furin cleavage sites. It's very, very common. Serious influenza viruses have them. In coronaviruses, they are all over the place. They come and go all the time in evolution. Okay, we've actually found some bat ones that have quite similar sites. So I think it's an evolution. I think it's a hotspot in evolution. It changes all the time. In itself, it means absolutely nothing. It appears to increase transmission of the virus. Okay, but I think no more than that. Also, one other point: if you use cell culture, so if you try and grow the lab in cell lines, you only ever lose the site. You don't gain it. Okay, so if it's a lab, if it's engineered in a lab, you would not. The result you're getting is the opposite. You actually get because you tend to lose in the lab. So I think it's a natural piece of coronavirus and virus evolution, and no more than that. It's not. There's no engineering going on that. I'm pretty certain of.
Got a couple on timing of release. So why did you and Professor Zhang not release the genome on January the 8th, 2020, once GenBank had already processed it? So we got the sequence on the 5th of January, and I released it on the 11th. Over that week, I didn't have the sequence myself until about less than an hour before I released it, okay? I was trying to get Professor Zhang to release it, but they were under great pressure in China not to release any data. Other groups had the sequence as well, and essentially there were instructions that there should be no publicizing of the outbreak. So we kept it to ourselves. We told the key people in China, right, people actually on the ground during the outbreak, but we were told not to release the data. As that week wore on, and I say other groups had it as well, as that week wore on, it became untenable to keep it to ourselves. As soon as they released it, as soon as they said it was a coronavirus, and the pressure grew online to release it, so it got to kind of breaking point. And I then persuaded Zhang to give me the sequence, and I released it. So it was that we were under. I think it's very
It's very hard to judge these people because the politics are very, very complicated there. Okay, and so it's not a simple thing. If it was a Western thing, it'd be different, but it's not so easy with dealing with China.
We had a couple of others in the room, and then in the grey jacket.
Have we understood the long-term impact of the virus infection?
No, I don't think. I mean, in terms of disease, no, I don't think we do. I think that there'll be a with this long COVID phenomenon. I think there are long-term implications of this that we don't know. We've learned an awful lot. I mean, the number of papers on COVID-19 is just unbelievable, right? My citations did really well last year, right? But there's clearly a lot more work needs to be done on that. I think it'll take years for this to kind of really come out, unfortunately.
The young lady at the front.
Can you treat coronavirus by using the inhibitor of the protein that it binds to?
Yeah. So there's a lot of work on antivirals for coronaviruses using drugs. I don't think they've got one yet that works that way. But most of the drugs that we've got, and none of them are actually that good at the moment, they're all old drugs that have been repurposed. Okay, they're drugs they had lying around they've used against coronaviruses, and sometimes they said they worked okay. At the moment, I don't think we have a really good antiviral that works. People have touted hydroxychloroquine and ivermectin. They do not hold up at all in clinical trial. So people are trying to do what you say. At the moment, the drug research hasn't really been as successful as the vaccine research. And the vaccine research has been spectacularly successful. Okay, so we do need an antiviral, like you say, but unfortunately, at the moment, we haven't quite got there.
Thank you so much. My question is, with the history on the wet markets, how did it take so long for it to spring over to humans? I mean, the last big pandemic that we can think of was, or that I can think of, is the Spanish flu. How did it take so long to kind of come over to humans?
Oh, no, people are exposed all the time, okay, all the time. If you go, they've tested people working in the market trade in southern China, and I think like 3% of them have antibodies to SARS-like coronaviruses, right? So people are being challenged all the time. 99 times out of 100, these things go nowhere, okay? They go absolutely nowhere. But, that one time they do. So this is one of the slides I didn't get to. SARS-CoV-2 is the 7th human coronavirus, right? 5 of those have appeared in the last 20 years, okay? And some of them have very similar stories to SARS-CoV-2. We have no idea where they come from. There's one called HKU1, okay? That was discovered in Hong Kong, Shenzhen, from an animal reservoir that's unknown, initially associated with pneumonia, and it has a furin cleavage site in the sequence. You heard of that virus? No. Right? That's now spreading. Luckily, now it turns out to be basically a common cold virus. So there's actually this emergence process is happening all the time. Absolutely all the time. Luckily, most of them don't go anywhere. Unfortunately, this one, the problem was, it's pretty transmissible. Also emerges in a big city in China just before Lunar New Year, okay? Which is the worst possible situation. And then you get millions of people then move literally the week after this, okay? And it's all over China very quickly. And because Wuhan's an international hub, it's out of Wuhan very quickly. That was the problem. So it's the worst possible place for it to happen, and unfortunately it happened.
I'll just say 2 more questions.
Yeah.
One from online, so I'll have to read that. Better put my glasses on. Why were the 4 studies on pangolin calves, why were they all released within the same 2 to 3 day period? May not know.
I can speak from our study. We had our paper published. We knew, so we'd heard, so we were working on pangolins, And then as our paper was being written, then as we heard that, there was a press release by another group claiming that they had a pangolin virus as well. I think it was just, I can't say any more than that. I wasn't involved in the other studies.
I get the last one. Hey, Eddie, it's Jackson Ryan from CNET. I'm not going to ask you about RATG13 or the miners or anything like that, but what I am interested in is you showed a lot of data about the virus sphere and how many viruses are out there. So really, my question, can we actually prevent the next pandemic? What do we need to do to be able to do so?
I think, well, the number one thing I think is better surveillance, absolutely better surveillance, right? And then data sharing. And unfortunately, those things didn't happen in this particular circumstance, right? And so, you know, it's become overtly political. It's become loss of face. All those sorts of things have meant that we've not been able to share data. And the political pressures, I mean, you know, like I say, Zhang has been under huge political pressure in China. And people, I don't think they really appreciate what it was like. And so the number one thing we need to do is building data sharing mechanisms so we have surveillance and that data is shared globally. That's the number one thing to do, okay? But we've got to cut through the politics, right?
Which we could all thank Eddie Holmes again. I'll just remind people we've got another, next presentation is on 19th of May when distinguished Professor Belinda Medlin from Western Sydney University presents Climate Change and Australian Vegetation. Where are we headed? We'll also continue to live stream all seminars and they will also be available to be viewed and on demand on our website, Chief Scientist and Engineer website. So I'd like to thank everyone for coming, particularly thank Fishburners for having us here. It's been fantastic and see you next time. Thank you.